All right, let's dive right in. Today, we are tackling the animal kingdom, and it is an absolutely incredible, complex, and sometimes, well, just plain bizarre world. So let's kick things off with a little puzzle for you. A sea sponge, a spider, and a shark. I mean, what could they possibly have in common? You've got the sponge, which is, basically just a colony of cells. Then there's the spider, with its creepy, crawly, jointed legs and that hard exoskeleton. And a shark, a super powerful predator. They seem worlds apart, right? But here's the thing. They all belong to one giant family, the animal kingdom. And it's not just them. Scientists have officially described over 1 million different animal species. And that's just what we've found so far. Think about that number. That is a mind-boggling amount of diversity to try and get your head around. So, the big question is, how on earth do you even begin to make sense of all that? It's kind of like being handed a library with a million different books. None of them are sorted, and each one tells a totally unique story. That's the exact challenge biologists are up against, trying to find some kind of order in all this beautiful chaos. And that's where we're going to start. We're going to look at how you sort a million species. You obviously need a system, right? A way to cut through all that variety and find the deep, fundamental patterns that connect every single animal. And the best way to think about this is that scientists are basically biological detectives. Seriously. But instead of looking for fingerprints or alibis, they're searching for fundamental clues. And these clues are hidden right inside an animal's basic structure, what we call its body plan. These are the clues that tell the real story, an animal's deep history and who it's related to. So what exactly are these clues? Well, let's open up the classifier's toolkit, so to speak, and check out the main features scientists use to crack this massive case. Okay, the first and maybe most basic clue is all about an animal's level of organization. Just think of it like a ladder of complexity, getting more advanced as you go up. So at the very bottom, you've got your sponges. They're really just loose collections of cells. Take one step up, and you find animals like jellyfish. Their cells have started working together to form tissues. Go another step, and you get creatures like flatworms, which group those tissues into actual organs. And then way up at the top, you've got animals like us, with really complex organ systems running the whole show. All right, next tool in the kit, symmetry. It sounds fancy, but it comes down to a really simple but super powerful question. If you could slice an animal, could you get two identical halves? The answer to that tells you a ton about how it lives and where it came from, evolutionarily speaking. And you can see here there are basically two main ways to slice it. On one hand, you have radial symmetry. Think of a pizza. You can slice it through the middle from pretty much any direction and get equal pieces. A jellyfish is a classic example. But then you've got bilateral symmetry. That's us. There's only one way to slice us, right down the middle, to get a perfect left and right mirror image. And this isn't just a small detail. This was a huge dividing line in evolution. It's what allowed for the development of a head, and with it, the ability to move forward with purpose. Okay, the last big clue we're going to look at is something called the phylum. I know, it sounds super technical, but really it's just a body cavity. It's the fluid-filled space between an animal's gut and its outer body wall. And believe it or not, whether an animal even has this cavity and what kind it is an absolutely crucial piece of the puzzle. And it turns out there are three main possibilities. First, you've got the acoelomates, like flatworms. Acoelomate just means no cavity. They're solid all the way through. Then you have the pseudocoelomates, which have a kind of false cavity that isn't fully lined. And finally, you have the coelomates. Yep, that's us again, with a true, fully lined body cavity. This was a total game changer for evolution because it gave organs a protected space to grow and get more complex, separate from the movements of the body wall. All right, so there we have it. We've got our three big clues: level of organization, symmetry, and the phylum. Now for the really cool part. Let's see how these few simple ideas come together to create this beautiful, logical map of the entire animal kingdom. And this blueprint right here, this is it. This is how it all works. You start on the left with, well, everything in the animal kingdom. The very first big split is based on that level of organization, which immediately separates the super simple sponges from everyone else. Then, the next big question is symmetry. Are you radial or are you bilateral? Boom, another split. Finally, for the bilateral animals, you sort them based on whether or not they have a body cavity. See how that works? It's like this elegant decision tree that takes all that chaos we talked about and organizes it perfectly into these distinct groups, which we call phyla. So now that we have our map, let's actually use it. We're going to take a quick tour through some of the biggest phyla and see exactly where they fit in this grand scheme. First up, the phylum Porifera. These are the sponges. And if you look at our map, you'll see they're the very first branch to split off. Why? Because they have the simplest body plan you can possibly imagine. They're at that cellular level of organization. That means no real tissues, definitely no organs, just a bunch of specialized cells all hanging out and working together. Okay, now let's hop over to a different branch on the tree, the Platyhelminthes. You probably know them as flatworms. These guys represent a huge evolutionary step up. They've got bilateral symmetry and they're at the organ level of organization. But, and this is key as our blueprint shows, they are acollimates. All their organs are just packed into a solid body. There's no internal cavity at all. Now, let's talk about the biggest group on the entire planet, the arthropods. We're talking insects, spiders, crabs, the whole lot. This one phylum makes up over 80% of all known animal species. Just let that sink in. Their wild success is all thanks to that tough exoskeleton and their super versatile jointed legs. And on our chart, they're way up at the top, true collimates with complex organ systems. And finally, let's bring it all home to our own branch on the tree, the phylum Chordata. So what makes a chordate a chordate? It's the presence of a notochord. It's a flexible rod that gives the body support. Now, invertebrates like you and me, we only have this notochord when we're embryos, and its job is to help guide the development of our backbone. So yeah, this is our spot. This is where we fit into the huge, sprawling family tree of life. Okay, so we've got this really elegant system for sorting animals. It's neat, it's logical. But so what? Why does any of this actually matter? What's the real takeaway here? Well, and this is the crucial point. This whole thing isn't just some dusty old historical exercise. It's a living, breathing tool that scientists use right now. The source material puts it perfectly, saying that the classification also helps in assigning a systematic position to newly described species. So when a scientist out in the field discovers a brand new species today, they use these exact same clues, symmetry, body cavity, organization, to figure out, okay, where does this new guy fit in the grand library of life? And that's because at the end of the day, this system is so much more than just a big filing cabinet for animals. It's really a map of evolutionary history. When we sort animals from the cellular level all the way up to complex organ systems, or from having no symmetry to having bilateral symmetry, what we're really doing is tracing the story of life itself. We're following that epic journey from the simplest of beginnings to the incredible, complex creatures we see all around us today. So this blueprint neatly organizes the million-plus species we already know about. But here's the kicker. Scientists think there are millions more out there, still waiting to be discovered. So I want to leave you with this thought. What new, undiscovered branches might we add to this tree of life someday? And what incredible secrets about the story of life are they just waiting to tell us?